last section, we have started about restriction endonuclease. As we know, restriction endonuclease, the enzyme, it is present in the bacteria. Bacteria it used as protection or the defense to protect against the bacteriophage. It is used to kill the bacteriophage. So it's a defense. So it is extracted from the bacteria. So at first, in the year of 1963, one of the scientists, his name is Arbor, he isolated first restriction endonuclease, means it is present in the bacteria, it is called E. coli. E. coli. They are using isolated two different types of restriction endonuclease enzyme. It is used to restrict the growth of bacteriophage. It is isolated from E. coli. So actually restriction endonuclease, it is present in it is under the class, it is called nucleases. It's under the class of nucleases. There are two different types of nucleases, exonucleases and the another one is endonucleases. So as endonucleases, as we know, by the help of this word only you can understand exonucleases. Exo means out. So exonucleases is used to cut the DNA, cut the nucleotides of both ends of the DNA. It is used to cut both the ends of the DNA. And the another one is endonuclease. Endo means it's inside. So it is used to cut the DNA inside, within the DNA, the nucleotides within the DNA. That's the different types of nucleases, that is exonucleases and endonucleases. There are different types of restriction endonucleases. Here you can understand some example. I will write ECO R1 and another one is HIN2. There are many restriction endonucleases. Now we have around more than 900 restriction endonucleases that is extracted from more than 200 strains of bacteria. So there are many. Here I have written two different types of examples of restriction endonucleases. First of all, we can understand how to name, what's the nomenclature of this restriction endonuclease, how to name the restriction enzyme. First letter, it belongs to genus. So it's E is belong to genus, that is Escherichia. It is Escherichia. And the another two letters that belongs to species name. So that is coli. So Escherichia coli. And this R, it's a type of strain. As we know, there are two different types of strain, S strain and R strain. So this R belongs to types of strain, and the one, it's a numeral, it's a Roman letter in which this bacteria belongs to. So this is the nomenclature. It is one of the important question for the both question as well as NEET. So this is the nomenclature of restriction endonucleases. Now we can understand by the help of the example how it can cut. As we know, restriction endonuclease is the molecular scissor. It's used to cut the DNA. How it can cut means, here you can understand particular restriction endonuclease, it can recognize particular sequence only. Particular sequence. The sequence contains 6 base pairs. 6 base pair. So it can understand particular sequence, that is 6 base pair. That sequence you can say recognition site. Recognition site. It is called recognition site. And this recognition site means it is forward. It contains 6 base pair. Forward and backward it will be same. So those sequence we can say, the another name it is called palindromic sequence. It is called palindromic sequence. I am writing one of the example here. It is GAATTC. This is one of the palindromic sequence for ECO R1. Example, ECO R1. So ECO R1 is a palindromic sequence is GAATTC. Means, and this is 5' and this is 3' and you can write the backward also GATTC. So this is 3' and this is 5'. Here you can understand forward and backward it will be same. 6 base pairs. That only you can understand palindromic sequence. As you know, you have studied about the palindromic sequence, there are many words like madam, forward and backward also same. Again you can see level, forward and backward it will be same. So those words you can say palindromic words. So here we are studying, it is called palindromic sequence. So it will be same. Now how this echo R1 can act this palindromic sequence. So it cuts between G and A. G and A, it will cut between G and A. Here also it will be G and A. So what will happen in the next point, in the next stage, it will be cut out. So GATTC. So here it will be, so this will be cut out, and here also GAATTC. So here it will be cut out. So this part will be separated and this part will be separated. So after that there will be a hanging ends will be formed. Hanging ends will be formed. Here it will be cut and here also. So here also it will be cut. So there will be the hanging ends will be formed. These hanging ends are called. These hanging ends are called sticky ends. Sticky ends. So by the help of restriction enzyme, we can cut. It can be cut. It is cut in the particular sequence, and it can be cut. So we will get the hanging ends. It is called sticky ends. Later, the sticky ends can be joined by the help of molecular scissor, molecular glue. It's a molecular glue. It is called DNA ligase. It can be joined by the help of DNA ligase. Here I have written one more example. It is heme 2. Heme 2 is extracted from Haemophilus influenzae. Extracted from Haemophilus influenzae, and it can cut one of the palindromic sequences GTC GAC. Here also it will be same GTC GAC. It cuts in between GC and GC. So it will get blunt ends. It is called blunt ends. And here it will be separated out. So hanging ends will be formed. That you can say sticky ends. It is called sticky ends. But here the same level it will be cut. There is no hanging ends. So this is called blunt ends. This is the difference between sticky ends and blunt ends. This is the explanation of restriction endonuclease. Thank you, students.